Protein trafficking. Although this topic it is related to the subject of microbial physiology, um, but here we will discuss it very briefly because when we are discussing the export of proteins, then uh, it is better if you have some idea about the structure of AFU transport system. If you have a look on the structure of outer membrane or inner membrane, you will realize that they contain different type of proteins that were first expressed in cytoplasm and then they are moved to the inner membrane or outer membrane and becomes the part of their structure. Even some proteins have been absorbed outside the cell. Now, the question is that how proteins that were once expressed or synthesized within the cytoplasm, how they moved outside the cell. So this question we are going to discuss very briefly. As I told you that when proteins they are expressed within the cytoplasm, they may have different destinations. They may have the fate to be integrated as integral membrane protein on the inner membrane. They have the destination in the periplasmic space, or they may be integrated on the outer membrane, or even if some microbial proteins or toxins, proteases, or other enzymes, they can be transported external to the cell. And how it is going on, we will focus on that topic. You have the idea that the protein that need export outside the cell, their movement it is facilitated by a specific signal molecule that is present on N terminal of the newly synthesized polypeptide chain. So this N-terminal signal sequence, it will interact with different chaperone proteins so that finally the protein will move outside the cell. And here briefly we can focus that how the proteins that having the destination outside the cell are within the internal membrane, how they are transported. Suppose कि एक प्रोटीन है जो कि न्यूली सिंथेसाइज्ड हुई है और इसकी डेस्टिनेशन है कि वो सेल से बाहर मूव करे, तो उसके लिए सेपरेट सिस्टम होगा दैट मे बी मीडिएटेड बाय अ स्पेशलाइज्ड chaperone proteins like the trigger factor. Trigger factor, it is one of the example of chaperone protein that will bind to the newly synthesized polypeptide chain if it is having the destination outside the cell. On the other hand, if the proteins it is having the destination to be integrated on the inner membrane, then a separate set of protein will recognize it and it may be called as signal recognition particle or SRP. If SRP protein will mediate the transportation of newly synthesized polypeptide chain, it means that it is going to be inserted within the inner membrane. Otherwise, if trigger factor or type of chaperone protein, if it will interact, it means that the protein it is going to be exported outside. For example, in this diagram, there are examples of two proteins, number 1 and number 2. If the protein it come in contact with trigger factor, which is a type of chaperone protein, then it means that it is going to be exported outside the cytoplasmic membrane, maybe into periplasm or external environment. After trigger factor, different type of chaperone proteins like the SEC-B protein or SEC-A protein, that are the example of chaperone proteins, they will facilitate its movement across the cytoplasmic protein into the periplasm or even outside the cell. On the other hand, if it is having the destination within the inner membrane, then it will come in contact with SRP or signal recognition particle and it will move or integrate it within the cytoplasmic membrane. So this is one of the example of GSP pathway, and commonly it is called as general secretion pathway. So if we have a look on different type of secretion system, then we will realize that there are several. There may be up to 7 secretory system for the movement of proteins. And each system will target a specific set of proteins. For instance, in case of gram-negative bacteria, they can be divided into secretion system 1 or type 1, type 2, type 3, type 4, and even sometime type 5 or so on. Or other some other branches of transportation pathways, they also exist. We can briefly have a look on the type 1 or type 2 secretory system. So this is the example of type 1 secretory system. Or generally it is also called as ABC pathway for protein export. ABC, it is from ATP binding cases. It is one of the component of transportation system. Actually, it is a protein that is present on the cytoplasmic phase of the cytoplasmic membrane, where ATP binds, and repeated cycle of ATP hydrolysis will move the protein outside the cell. So it is having a complete channel from the outer membrane to inner membrane, and this channel is continuous from this periplasmic space, so that there is a continuous channel for the transportation of the target protein. So this is type 1 secretion system. In addition to that, the most common example of the secretory system, it is the type 2 secretory system, and it is the main branch of general export pathway. And here the target protein that is having N-terminal signal sequence in sec-dependent manner. Sec-dependent manner means that some chaperone proteins, they are involved in the transportation from cytoplasm to the periplasm. So the movement first, it is sec-dependent manner, where chaperone proteins, they are involved. When protein it is transported to the periplasm, then the N-terminal signal sequence, it is cleaved. After that, the protein in the periplasm, it is folded, and after folding, it is transported outside the cell. So like the type 1 secretory system, this system also mediated by the repeated cycle of ATP hydrolysis. Now finally we can say that there may be different type of secretory system. We have just focused on 2 systems, and each set of system, it transport different type of proteins.